Hi, this is our presentation on the effect of alkalinity on plant growth. My name is Jenna Bly.
I'm Cassandra Lewis.
I'm Jessica Collins.
And I'm Sophie Negri.
Basilovocalis fabaceae, also known as the common bean, was utilized to investigate the effects of abiotic conditions such as soil pH on plant growth. The optimal soil pH range for beans is 6 to 6.5, within which all nutrients are available for uptake. However, increasing the pH changes the nutrient availability of different minerals and therefore can result in deficiencies of nutrients that are limited. Nutrient deficiencies of which lead to a decrease in plant growth and survival. Consequently, leaf chlorosis in the upper leaves is a result of decreased chlorophyll content in response to iron deficiencies that occur in alkaline soils. As pH increases, temperature stresses decrease, which can reduce the loss of chlorophyll content and therefore the treatment plants appear greener. Alkaline soils also alter root morphology, reducing root systems and limiting plant access to nutrients and water. The aim of the experiment was to determine if increasing alkalinity within the growth medium would decrease the seedling height, root length, weight, and chlorophyll content. As a result, it was hypothesized that with increasing alkalinity, seedling height, root length, weight, and chlorophyll content would decrease. The null hypothesis would indicate that no change in the variables between the alkalinity and control group.
Materials required included bean plants, water, pH 9 solution, nutrient solution, and measuring equipment. The method was divided into two parts, being the creation of the NaOH solution and the monitoring of bean species over the course of the experiment. The solution was made by the addition of small increments of NaOH to water, with consistent testing of pH to reach the desired alkalinity. The second part of the method was the data collection from bean plant samples. Each specimen initially received nutrient solution, followed by 20 milliliters of the allocated treatment. Initial plant length was measured, and the samples were then stored in a glasshouse environment. Alternating treatment patterns then ensured an experimentally blocked design. Measurements and treatment watering continued every 3 to 4 days until the conclusion of the growth experiment. At the conclusion of the experiment, bean plants were removed from the potting mixture and root length, segment weight, and chlorophyll content was determined.
Having a high pH of 9 within soils can impact the nutrient availability for plant growth. Nutrient availability had clear impacts on how plants physically grew and influenced visual characteristics. Visual observations found that there was a minimal difference between the growth of both groups. This can be seen to the right of the slide with all 8 plants at a similar height. In contradiction to the original hypothesis, there was a significant difference in greenness within groups, treatments indicating a higher SPAD unit. This has likely occurred due to an uneven balance of nutrients presenting as yellowing within older leaves and lightening of younger leaves as seen in figure 6. The treatment group then demonstrated a significant reduction in root growth as compared to the control. This can be seen in figure 5, whereby the root growth is clearly stunted in the treatment group. Finally, there was an overall reduced biomass observed between groups, with significant differences in root biomass as previously highlighted, as well as slight differences in shoot biomass. As a result, the treatment group was potentially under greater levels of stress and therefore became stunted.
Statistical findings determined that there was not a significant difference in growth rate. The one-tailed p-value of 0.22 only suggested alkalinity had an effect, with a conclusive finding not being reached. Mean root length of the control was greater. With a one-tailed p-value of 0.016, it was determined alkalinity had a significant effect on root growth. All sections of the plant weight, including shoot, root, and whole plant, had significant p-values. Final weight findings concluded that alkalinity treatment had a significant effect on the reduction of plant biomass. The SPAD chlorophyll greenness testing was significant, but contradicted the hypothesis of greenness reduction in the treatment group. With a one-tailed p-value of 0.037, the greenness of the treatment group was significantly higher than that of the control.
Soil pH can impact variables such as soil structure and nutrient availability, each affecting plant development. Certain nutrients such as phosphorus, potassium, and sulfur become more available in alkaline soils, while others such as calcium and nitrogen become less available. When pH is outside its optimal growth range, nutrients become tightly bound to soil and are no longer available for root uptake. The hypothesis was partially supported, with root length and weight of the treatment plants being reduced comparatively to the control. However, the alkaline treatment plants demonstrated more greenness or chlorophyll function than the control, opposing the hypothesis. This may be due to the change in phosphate availability and stronger nutrient binding to the soil. It is important to note that while collecting the root and plant wet weight, it was assumed each plant had the same amount of sand still attached. It was also assumed the initial root weight was equal. Plants can take between 3 to 6 weeks to show any symptoms of environmental factors. Therefore, a longer time would be needed to identify all effects of the alkaline soil. After 3 weeks, the leaves of both the control and treatment plants had begun wilting. Temperature variability was not accounted for when watering, potentially causing excess stress on samples. Due to a lack of resources, lab analysis to determine reasons for some symptoms of the treatment plants was not conducted. Future experiments could include a range of pH, investigating a variety of basic and acidic solutions. Any future experimentation should aim to reduce these limitations.